So what I will do in my presentation, I will describe how our laboratory searches for differences between the human prefrontal cortex and the prefrontal cortex of other species, and how we investigate when, where, and how these differences occur during development. And as my two esteemed colleagues, Javier and Huyeb, described eloquently, there are several reasons why we should pay attention to prefrontal cortex. It plays a critical role in human cognition and behavior, and it occupies actually the anterior 1/3 of the cerebral cortex in humans, other apes, and monkeys, and as you can shown here in blue, and it sits exactly in front of the motor area shown here in red, and what it has a specific connection, as Hugh explained, with the medial dorsal nucleus of the thalamus. However, unlike anthropoid primates, in all other species so far analyzed, regardless of how big is their cerebral cortex, the prefrontal cortex occupies this medial part that Hugh described, and it's actually agranular, and while the motor areas occupy the most anterior part of the lateral frontal cortex. And so what I will do in the rest of the presentation really focus on two major questions. One is, does human prefrontal cortex have cell types that are different from humans and other species? And we'll compare that to our closely related primates. And then in the second part, and most of the story will be about the second part, is when are the areal identity, the size, and the pattern of connectivity of the prefrontal cortex specified during the development. And in this second part, I will actually summarize recently published papers. And it's work led by Mikihito, two very talented postdocs, Mikihito Shibata and Kartik Pattabiraman. Kartik just started his own lab at Yale Study Center, and I cannot recommend him more enthusiastically as a mentor if anybody is looking for a job. And so what Mikihito and Kartik, together with several colleagues here listed, is actually to identify a previously unknown role of retinoic acid in regulating the areal identity, size, and connectivity of prefrontal cortex. So the first obvious question is, do humans have cell types in prefrontal cortex that are different from other closely related species? And to address that question, Andre Sosa, Jay Ma, Mario Škarica, and Ying Zhu over the last several years have been using a single-cell RNA-seq, in this case particularly single-nucleus RNA-seq. So what they did is they isolated nuclei from fresh frozen marmoset brain, which is a New World monkey, macaque, Old World monkeys, chimpanzee, our closest living relatives, and humans from the dorsolateral prefrontal cortex, and then basically in unbiased manner using a single RNA-seq, look at the gene expression, and then basically they can use that data set to basically cluster the cell types in agnostic manner. And basically what they identified, 113 transcriptomically defined cell types, including 40 excitatory, 45 inhibitory, and 28 non-neuronal. The important take-home message here is that all neuronal cell types, both excitatory and inhibitory, were present in or shared between human, chimpanzee, and macaque. That was not true for marmoset comparing the two other three. There were examples where marmoset does not have some cell types as well, that there was a cell type that marmoset has, but the other three primates don't have. The second important take-home message is that there was only one cell type found exclusively in humans. That's a type of microglia. This microglia has also recently demonstrated by another group, indicating that it's probably truly human-specific. It's also present fetally, so probably not affected by our environment and diet and many other features. And in addition, also, when you look at the relative proportions of the cell types that are shared, you can see they're actually relatively similar. There were slight changes in only several, including two subtypes in upper layer intratelencephalic projecting neurons. But overall, the relative proportions of these cell types between human, at least a chimpanzee, were very, very similar. But the main differences between the four species that all cell types, including those that are shared, exhibited both gain and loss of human-specific gene expression pattern, as shown here in bar graph. So the gray is the gain, and the black is the loss of human-specific expression. So basically, even though the same cell type that exists in human and chimpanzee or human other primates have exhibited a human-specific either loss or gain of a gene. So indicating that they all have changed regardless of their identity. And so the next question we wanted to address, and I'll spend most of my presentation on this question, is how are the areal identity, the size of the prefrontal cortex, in particular the expansion in anthropoid primates, as well as the pattern of the connectivity specified during the development. And so I will summarize almost 15 years of work, basically telling you how we get. And so the first question is, how early do we see areal differences in the primate and human cortex, and when can we see the transcriptomic signatures of the prefrontal cortex? And so over the last almost 20 years, our lab has been using genomics, and so we've been following a gene expression across the entire human development in humans and other non-human primates, particularly macaque. And so what we observed, that the first areal differences can be observed as early as the beginning of the second trimester. That's around 12 weeks of post-conception. And if you look at the human brain, actually it's very small. It's between 1 to 2 centimeters wide. I mean, that's like this, okay? Most of your neurons are actually not present, actually. And even in the cortex, if you look carefully, this is a cortical plate. You can see basically that most of the neurons are still migrating, actually. You can see this lot of migration to the subplate and fetal white matter. And if you look at the organization of these cortical neurons in the cortical plate, they are actually bipolar. They don't have dendrites. They don't have any synapses. And actually, most of the synapses and input from thalamocortical, as actually many of you have shown here, is present within what's called the subplate. And there are no yet intracortical synapses. And what it is really also to demonstrate there are no laminar differences. This is why it's called cortical plate. And if you compare, for example, this is a future superior temporal cortex and this is a prospective dorsolateral, you can see that actually these neurons are immature, jam-packed, and just based on NISL, it's very hard, almost impossible to see any areal differences. And so historically, we have been interested in these 11 areas that are areas of the prospective neocortex, 4 areas of the prefrontal cortex: primary motor, primary somatosensory, primary auditory, primary visual, as well as some subcortical that I'm not going to talk today. And what was really important is that early on, this was almost published 15 years ago, we actually observed a dramatic differences across these prospective areas. Even though most of cortical neurons are not yet even generated and they are still migrating, we could see basic transcriptomic signatures that can divide these areas, these dissected areas, into future prospective areas. So you can see that most of the gene expression pattern comes from the areas that will be in the frontal part, as well as those that are in the temporal. And you can use this gene expression pattern, which tells you that basically these neurons know that they will be in the prefrontal cortex versus, let's say, temporal cortex. You can also see actually perisylvian areas that belong to frontal, parietal, and temporal cortex, that they have some of the same signatures. And then you can use this to cluster areas during early and mid-fetal development, and you can get a really very faithful recapitulation of areal topography in the adult cerebral cortex, which goes in frontotemporal axis. So you can see that areas of the prefrontal cortex cluster together. This is a primary motor, somatosensory, parietal, visual, and then all the way to the temporal, including association temporal areas. Why is this important? Well, this tells us 2 things. One is that neurons already know that they will be destined prefrontal cortex versus temporal. These gene expression changes happen at the level of immature neurons. This is not at the level of progenitor, just to classify this. Anyway. And so we can now look what distinguishes, for example, neurons that are becoming a prefrontal cortex from those that will be in, let's say, parietal. And then we can look at the gene expression and we can see, are there any categories of genes that are selectively or significantly enriched. And then when we look at this, actually, we observe several categories. First, as you would expect, because you are developing the circuits, you can see that the genes that are related to synapse organization, formation of synapses, axon development, as well as excitatory projection neurons are enriched in cells that will become a prefrontal cortex. But in addition to these really generic categories, we also observe the genes that have been classically connected to retinoic acid signaling have also been enriched in neurons of the prefrontal cortex. And here is a couple of examples from our 2009 paper. So, for example, genes that are important for transmitting retinoic acid signaling, such as the classical receptors such as RXRG, RAR, beta, are upregulated in prospective prefrontal cortex, like MACE2, cerebellin-2 are also known targets of retinoic acid in other parts of the nervous system. Historically, this has been studied for almost 40 years. And then what is also important, we observed almost 15 years ago that a gene that, and this is an important part for the rest of the presentation, called CYP26B1, this is a gene that encodes an enzyme that degrades retinoic acid, was selectively enriched in primary motor cortex, and we'll show you that this is an important mechanistic insight into it. So what is the retinoic acid? So retinoic acid is a small molecule. Actually, it's one of the first, I think it's the first molecule that have been shown to be important for patterning of the central nervous system, has been classically associated with the patterning of the caudal part of the nervous system, neural tube, the spinal cord, but in this case, I'll show you the evidence that it does this also in prefrontal cortex. So retinoic acid is generated from vitamin A, that's why you need vitamin A, and cells that express one of the three enzymes, LDH, A1, A2, and A3, can make retinoic acid, which is then released. And then what it does is actually it binds to a nuclear receptors that there are two categories of RAR and RXR, A, B, and G. And so they can form heterodimers, and together with retinoic acid, they actually bind the retinoic acid responsive elements, and they can drive the gene expression pattern, and has been shown to be important for cell fate specification in other parts of the central nervous system, as well as other organs in the body. But also what is important that this signaling is controlled not just by enzymes that make it, but enzymes that and the genes that this will stop working. It's also important that there are three other enzymes which are called CYP26, A1, B1, and C1. These enzymes, cells that express these enzymes are not responsive to retinoic acid, and actually what they do is they degrade the retinoic acid. So by controlling which cells make it, which cells are responsive, and which cells degrade, you can create a spatio-temporal gradients of retinoic acid, and that has been shown to be a very universal mechanism by which you can pattern organs during embryonic development. And so first, what we wanted to test, since we see genes that have been previously linked to a retinoic acid signaling to be upregulated in early fetal and mid-fetal human cerebral cortex, we also observed this pattern in a macaque monkey. We wanted to see if there is a retinoic acid there, so we used two test assays. One is the ELISA assay, as well as using a RA-Relaxi reporter cell lines. I'm not going to show you that, but and so when Mikihito and Kartik measured, this is from fresh human samples. This is a mid-fetal development. You can see that the prospective areas of the prefrontal cortex, even though there are no areal differences, there are no laminar differences, neurons are still migrating, and there are no major gyri. Basically, you can see that the areas of the prospective prefrontal cortex can be identified by actually upgraded, elevated levels of retinoic acid. This also forms a really beautiful and consistent gradient of going from medial prefrontal areas to the lateral, and then once you reach what will become a prospective motor cortex, you see that this gradient is diminished. And this is a schematic. We also see a little bit in a temporal association areas. We actually have identified a human-specific signaling there. I will not talk about it, but I'm happy to talk about it in discussion. We look at macaque monkey at equivalent embryonic age. We see the same gradient, with exception is that the levels of retinoic acid, this is a fresh samples, were slightly lower in macaque comparing to human. And then we also look at mouse, and we observe the retinoic acid to be generated or present within what will become what it corresponds to. This is at embryonic, this is at P0, which is equivalent age to early mid-fetal development in humans. And we could see retinoic acid only in the medial part that you and others have described is a mouse medial prefrontal cortex. So clearly there is a homology between medial and prefrontal cortex in primates and mice. So the next question, where do we get this is at the level of molecular, what makes this retinoic acid? And so we look at the three enzymes, and we found that two of them, A1 and A3, are ubiquitously broadly expressed in the mid-fetal prefrontal cortex, medially, dorsolaterally, orbitally. This is true for macaque. And when we look at the mouse, we could only find, and actually this was discovered almost 20 years ago by Ursula Drager. She found that one of these enzymes was expressed here. A3 is expressed in medial prefrontal cortex, but A1 was not expressed. So clearly there are examples of species-specific changes in the expression of the key synthesizing enzymes. The next thing we wanted to test what is the role of retinoic acid in doing that, and we actually turned to mice, and again, mice have the retinoic acid present developmentally in the medial part of the frontal cortex. Then we look at the expression of genes that encode nuclear receptors, and we look at in human, macaque, and mouse, and we observe that two of them, RAR beta and RXRG, have a anterior to posterior gradient of expression pattern. This gradient was present also in mouse. So we knocked down these two genes. Actually, these mice are alive, and then we cross them to rare LAG-Z mice generated by Janet Rossant. And so these mice are transgenic mice that actually have a retinoic acid reporter driving LAG-Z. So the cells that express LAG-Z are responsive to retinoic acid. So we can say which cells in the mouse brain are responsive. And as you could see, the cells in the medial part, which will become future prefrontal cortex, are responsive to retinoic acid, and this is diminished in a double knockout. We also identified third receptor, may just triple knockout, so we'll find out soon whether there is a total abolishment of retinoic acid. So we wanted to see what are the effects of attenuated retinoic acid signaling on these cells. So the first questions we did, we actually look at the gene expression, because if you can remember, told you earlier that work from many colleagues have shown in other organs as well as other parts of the central nervous system have shown that embryonically retinoic acid is a powerful mechanism that controls the gene expression, specifically gene expression related to cell fate specification. And so we actually microdissected motor cortex, medial prefrontal cortex, and orbital. You could see that they are clearly separated in wild-type mice. These are littermates. But when we look at the double knockouts, the differences in gene expression within these three areas became diminished. Then we look at what are the genes that are differentially dysregulated and in the medial prefrontal cortex, and we found that actually they are related to, again, similar categories of genes that were also upregulated in embryonic and fetal human prefrontal cortex and macaque. So those are genes related to synapse development, glutamatergic synapse, and neuron projections. So we wanted to test whether there are any changes in connectivity. And so, as some of you already may know, this is that prefrontal cortex, at least in primates, have more dendritic spines than the neurons in posterior cortical areas, creating these anterior to posterior gradients of dendritic synaptogenesis. And so we wanted to test, are there any changes in the density of dendritic spines in these mice? So these are the double knockout mice that have lacking two critical receptors. And we saw a moderate but significant reduction in the number of dendritic spines in prefrontal, both upper and deep layer pyramidal cells. This was not true for all other non-prefrontal areas that we looked. There were no differences indicating that this phenotype is specific for prefrontal cortex. And then we look at actually connectivity between prefrontal cortex and mediodorsal nucleus using two techniques. One is a DTI tracing in mice, as well as the viral retrograde labeling. And you can see that basically that projection from reciprocal projection from mediodorsal nucleus and medial prefrontal cortex are attenuated or at least reduced in this double knockout, indicating that loss of retinoic acid has a consequence on both dendritic spine formation as well as formation of long-range connectivity between mediodorsal nucleus and prefrontal cortex. So what does mediate this? Can we identify those? And one of the genes that caught our attention was cerebellin-2. It's a gene that encodes a synaptic organizer, have been shown to be important for forming synapses. Also was one of the genes that we actually identified in 2009 is a gene that really labels prospective prefrontal area. So this is a expression of cerebellin-2 in mid-sagittal human fetal brain, and you can see that expression is high in areas that will become a future prefrontal cortex and goes down when it comes to medial motor cortex. These are coronal sections in macaque. You can see also high prefrontally, low posteriorly. And in mouse, even though it's called cerebellin, it was actually identified as a cerebellin-specific gene. It's actually not specific. You can also see it in what will become a future prefrontal cortex in mice. In addition to big areal differences in between expression of cerebellin, we also observe laminar differences. In human mid-fetal development, cerebellin-2 is expressed through entire cortical plate, including upper and deep layers, while in macaque monkey as well as mice, was predominantly expressed in upper layer and was not expressed at high level in deep layer, specifically in layer 5. So there are, in addition to areal differences, there were also laminar differences. And as I mentioned, this was also gene of interest because has been linked previously by many groups to basically be important synaptic organizer by linking urexin and glutamate receptor, and this complex of protein has been also linked to several neuropsychiatric disorders. And so to identify and link this to retinoic acid, we actually wanted to see if there is any mechanistic connection. And so Sidney Muchnik, a talented grad student in the lab, identify an enhancer. These are not coding elements that gives you a spatiotemporal specificity of gene expression, both in mouse and human, that is only present during the development in prefrontal cortex, but not in other areas for which we had data. This enhancer, if you take a human enhancer and now express a LAG-Z in a mouse, you can see that the expression of LAG-Z is expressed in both medial as well as the frontal pole. Recapitulate what we see in humans. And this enhancer had multiple binding sites for retinoic acid receptor, which was very exciting. And so we wanted to see whether cerebellin is directly controlled, and we were able to show that it's induced by expression both of RXRG and RAR beta, as well as there was a significant loss of expression and double knockout, as you would expect. And then here is the lucky single most important factor in life. Miki Kito runs into my office saying, Nenad, I have to show you something. And in addition to looking for retinoic acid, he also observed that there are cluster of sites that are predicted to be bound by a transcription factor called SOX5. And what was important is Mikito identified two hominini-specific deletions, 1 and 2, that were deleted and are purified in human populations. So none of us have this, at least to the almost 100,000 genomes. And what was very exciting, very lucky, single most important factor is that these sites are predicted to be controlled by SOX5 and work from our lab and Jeff Mack's lab shown in 2008. SOX5 is a critical transcription factor for specifying layer 5 and 6 excitatory projection cells and formation of corticothalamic and corticospinal projections. So here is expression of SOX5. It's highly enriched in deep layer. There are some expression in upper layers. And this is the expression of cerebellin-2 in neonatal prefrontal cortex. You can see it's mainly restricted to upper layers. So we hypothesize that basically the reason you don't have expression in deep layers like you see in humans is that basically SOX5 can bind this enhancer and repress it in non-human species. And so to test this, Mikihito he looked at the expression of cerebellin-2 in SOX5 knockouts. These are conditional knockouts. They survive. And now you can see that basically endogenous cerebellin-2 is expressed also highly in layer 5 and 6. And to test this hypothesis, he actually engineered a mouse by replacing a mouse enhancer with a human enhancer. Remember, this enhancer does not have these clusters of SOX5 binding sites, and the clusters are more efficient than individual sites, just to give you a hint. And also, you can see that basically by having even though this laser pointer is about to die, you can see that basically even in the context of SOX5, which works as a repressor, we have found it in 2008, if you don't have these binding sites, SOX5 cannot repress, and there is expression of cerebellin to mimics what we see in the human prefrontal cortex. Are there any consequences of this human-specific expression? And so to do that, Mikihito and Kartik actually did retrograde tracing of callosal as well as the extratelencephalic layer 5B cells and showed that basically there is a increase in dendritic spines. We assume that this is synaptogenesis. We actually did EM of both upper and deep layer in response to increased cerebellin-2 expression. So I hope I was able to show you that this is important for controlling gene expression of genes that are essential for making synapses. But how do you control this gradient? And so hint came also from our initial 2009 paper. So as I showed you earlier before, is that we have found that many genes that have been shown to be controlled by retinoic acid, including cerebellin, to which we were able to link specifically in the cortex to retinoic acid, are enriched in what will become future or prospective prefrontal cortex. But what we observed that a gene called CYP26B1 was expressed by prospective primary motor areas. And so we hypothesized that this could be what controls this gradient. And we showed that something similar exists in mice. So future anterior insula and motor cortex have this stripe. This is at P0, and while the cerebellum 2 is expressed in front of it, and you can see this is a anterior view. So you see green, which is a mainly orbital and medial prefrontal cortex, green and red, which would be motor areas, and then blue is future primary somatosensory area. And you can see that this gene is expressed in a gradient, high in motor, low in prefrontal cortex. So we hypothesized that this is what is preventing spillover of retinoic acid. So we made a conditional knockout, we made a knockout whole body, which actually unfortunately died at E18.5. We just made a conditional that is alive, so we'll know a little bit better. And so you can see the retinoic acid has a high level in medial, but low levels in secondary and primary motor. There is a beautiful medial to lateral gradient. If you knock out a gene that is present in prospective motor areas, the levels of retinoic acid go up. There is a still gradient, but now the motor areas have levels that you would see in the wild-type prefrontal cortex. And we also see induction of cerebellum 2 or upregulation of MACE2. These mice also have this lateral bulge, it's 100% penetrant, and also we don't know what it is. We are trying to trace this lateral bulge. And we also then trace from medial thalamus at E18.5. We did this by putting dye crystals. You can see some projection. They are just arriving to the frontal cortex. But in the knockout mice, we see three things. One is exuberant and precocious, as well as ectopic projection, not just in medial, but also a lateral part, which will be a motor cortex. So here is the model. Future prefrontal areas can be mapped or demarked by expression or this retinoic acid gradient, which is controlled by an expression of CYP26B1 expression in anterior insula and motor cortex. Knocking out this leads to expansion of this retinoic as well as expansion of medial dorsal nucleus, and we were able to link this to at least one gene that is important for synaptogenesis. Thank you.